Hello, my AP Biology students. For this presentation, we're going to talk about RNA, the crazy member of the nucleic acid family, also the oldest. RNA stands for ribonucleic acid. So it's very similar to deoxyribonucleic acid. In fact, the only real difference is that ribonucleic acid has one more oxygen. Deoxyribonucleic acid, so ribonucleic acid has one more oxygen. That's important because oxygen is a very reactive chemical, which makes RNA less stable, which actually matters for a lot of different reasons that we'll get into as we move forward. Ribonucleic acid is very similar to DNA in structure. It's a polymer made of monomer units. The monomer units are nucleotides, which are very simple. The nucleotides have three main points, sugar and a phosphate, okay? And you can actually see on the diagram to the right, the circled portions, you've got the two sugars, the 5 carbon chains making up an RNA backbone, and the phosphate, which is the yellow and red structure that's been circled. All right. What happens here is that the RNA exists very similar to DNA. It has a sugar phosphate backbone, and it has nitrogenous bases that point to one side. Now, DNA has this double helix structure where the nitrogenous bases point at each other and complementarily base pair. Well, RNA can also complementary base pair, but it doesn't actually exist as a double helix. It usually exists only in a single strand. A lot of times that single strand doesn't complementary base pair with something else, not for any length of time. And if it is complementary paired, it's usually because the strand has folded into some sort of structure. Okay? Now, there's one huge difference between DNA and RNA, and that huge difference is right here, okay? RNA, no thymine, okay? There is no thymine in RNA. Instead, there is a nucleic acid known as uracil, okay? Uracil is a replacement for thymine. It's a precursor. Again, it helps contribute to the lack of stability in RNA and all of those things. So RNA still has adenine, it still has cytosine, it still has guanine, but instead of being ATCG, RNA is AUCG. Now, I'm going to tell you right now, when we talk about how we get from DNA to a protein, where the ATCG ends and the AUCG begins is typically one of those points where people find confusion. So you really need to get this in your head. Is it RNA? If it is RNA, there is no T. Is it DNA? If it is DNA, there is no U. All right? RNA, no thymine. RNA, uracil. Now, RNA, with all of these awesome and amazing traits that we've been talking about so far, comes in a multitude of forms. Okay, first things we're going to talk about, what's known as mRNA or messenger RNA. And this will make more sense earlier or later on, but it also includes pre-messenger RNA or primary transcripts. Messenger RNA is RNA that's generated when DNA is transcribed. You can see this over here. Here you got a DNA strand. Over here to the right, you got a DNA strand. And right here, a strand of RNA is being built. And it's being built by complementary base pairing to the DNA strand. We're going to talk about what this means as we move forward, but that's where messenger RNA or pre-mRNA or primary transcripts come from. All right, they are based off of DNA templates. So messenger RNA is generated, and the process is known as transcription. You're transcribing DNA. So that's one type, mRNA, or messenger RNA. Next up is tRNA, or transfer RNA. All right. Now, tRNA is a single-stranded molecule, just like messenger RNA. But this structure right here is tRNA. Now, it looks like it's a mess of who did what where now. But if you look at this diagram down at the bottom, which is sort of how we like to describe it, what you've got is one strand that starts here and ends here. And what you've got going on is complementary base pairing. And that's actually what has this structure. Whoa, why is my pen doing that? That is what actually has this structure held together in its shape, is the complementary base pairing gives it its shape. Okay? It's what I said before. It's not always double-stranded. This is actually one strand. So this structure right here is actually one strand. All right? Now, transfer RNA, or tRNA, is a single-stranded molecule, folds into this structure. And remember, structure equals, you better have said function. All right? And that structure allows it to bind to two places. Down here, there's something called an anticodon, which will bind with messenger RNA. And up here is a tail that will attach to different amino acids. Amino acids are the monomers that make up the polymer polypeptides. All right, so there is another type of RNA, but to get into that, we need to talk about an organelle that exists in cells. This organelle is called ribosomes. Ribosomes are a structure that is made of protein and RNA. And what it does is, ribosomes are the site of protein synthesis. So ribosomes are where your cell makes its polypeptides. Ribosomes are where your cell makes its enzymes. Every structure that your cells have, every function that they do, comes from the ribosomes. All right? And as we get into this process of protein synthesis, we're going to talk a lot about how ribosomes do this. But let's just talk a little bit about the basic structure here. Ribosome has two pieces, a large subunit and a small subunit. If you take a look at this diagram right here, what's on this side that's colored purple, dark purple, light purple, gray, that's the large subunit. And what's on this side that's colored two different shades of blue is the small subunit. And during the process of protein synthesis, they combine to make a ribosomal structure. Ribosomes are made of protein themselves. That gives us a little chicken and an egg problem, but we'll talk about that later on. Ribosomes are made of proteins themselves, and they are also made of a substance called rRNA, or ribosomal RNA. Now, ribosomal RNA actually exists inside the ribosome at places where the ribosome performs its function. It's actually a very integral component to the process of protein synthesis. Okay? So we've got three types of RNA: mRNA, messenger RNA; rRNA, ribosomal RNA; tRNA, transfer RNA. Messenger RNA is made during the process of transcription. Transfer RNA is used during the process of protein synthesis, or what we call translation, which is when amino acids are joined together. And ribosomal RNA is key to both of those processes because it is an important structural component of ribosomes. All right. Do not confuse ribosomes with this. We've got this weird thing. Enzymes are biological catalysts. They are substances that allow living organisms to have their chemical reactions happen more quickly. If the chemical reactions happen more quickly, then the organism can become more complex. It can live. It can survive. But enzymes are protein structures. Protein gets their structure from DNA. But you need enzymes to use the DNA. Where do those enzymes come from? How did that happen on an evolutionary scale? And the answer turns out to be part of the reason that RNA is so interesting. We mentioned before that RNA is not quite as stable as DNA. So as a long-term generational information storage molecule, it's not as effective. But in the short term, when life was just evolving on this planet, RNA was useful to store information. And it was useful for another reason, because RNA can actually function as a mild enzyme. And that is where we believe the process of transcription, translation, DNA replication, reverse transcription, all arose. It arose because RNA was originally the storage molecule, and RNA catalyzed its own copying. It catalyzed its own use. And then some change happened. DNA became the useful molecule. And until these early enzymes were generated to begin this whole process, RNA was used to catalyze the use of DNA. We call the structures that allow this ribozymes. Dun, dun, dun. So like I said, I went through this whole thing about, RNA, DNA, the enzymatic property. The key here is for you to take away that RNA can act as an enzyme and as an information storage molecule. We believe that that's how RNA for early organisms held the genetic information. When DNA, which is more stable and henceforth better for, passing your information on from generation to generation, became more common, RNA took on a catalyst nature. We actually think that's part of the role it plays as rRNA inside your ribosomes. All right. And ribozymes, which still exist in certain organisms in certain places, are evidence for the evolutionary relationship between RNA and DNA. And that's it for our second presentation. I hope you all enjoyed.